Bismillahirrahmanirrahim. In the name of Allah, the most merciful, the most beneficent. Today we are going to discuss assessment of polyphenolic contents and in vitro wound healing activities of Syzygium aromaticum or Caryophyllus aromaticus. There is clove. The outline, it's a study which we conducted in our lab. The outline of the study of the presentation are introduction, objective, work plan, methodology, research and discussion, and with the conclusion. Introduction. Why herbal medicine? Herbal medicine, according to the World Health Organization, almost 80% of the world population relies on herbal medicine for primary health care. Herbal medicines are used to treat diabetes, aging, wounds, infection, asthma, hypertension, cancer, and number of other diseases. Why wound healing? Wound is the third major reason of medical visits in developing countries after heart diseases and cancer. Here we are supposed to discuss different phases of wound healing, which are overlapping phases, and what are the factors or what are the parts of skin which are associated in that specific phase. First of all is hemostasis, which is blood clotting or blood clot formation. Second phase is inflammation. Inflammation is the response of the body to painful stimuli. The scab is formed. Fibroblasts are active in inflammatory phase. Macrophages are active. Blood vessels are actively supplying the blood. Then third phase is cell proliferation and migration. The, in that phase, fibroblasts are proliferating and subcutaneous fats are active. And the fourth phase is remodeling. Freshly healed epidermis is then converted to normal skin. What are the factors which cause delay in wound healing process are uh, local factors, which are infection, size or extent of the wound, and oxygenation. And there are systemic factors. There are age, obesity, nutrition, immune status, and other diseases. For example, diabetes. Objective of the present study was to evaluate the in vitro wound healing activities of Syzygium aromaticum or Cryophyllus aromaticus, identification of bioactive compounds responsible for these activities, and the quantification of polyphenolic compounds responsible for wound healing activities of Syzygium aromaticum. First of all, we collected Cryophyllus aromaticus or Syzygium aromaticus flower buds from Ayub Agriculture Research Institute, Faisalabad, Pakistan. We ground the samples and made the extracts from the sample. Then we cultured human cell line. For this purpose, we cultured human oral epithelial cell line H400 in a 12 well cell culture plate. Then we created a wound using 220 to 200 microliter piper tape, and the size of the wound or the size of scratch was 1 millimeter. Treatment of wound with plant extracts, and then we analyze the effects of extracts using statistical analysis. We also performed HPLC analysis for the extracts. For extraction of Syzygium aromaticum, the powder was dissolved in 50% ethanol, placed in a shaking incubator, vortexed the solution, and filtered. Uh, that filtered solution was used for both HPLC analysis, and it was evaporated and re-dissolved in PBS for human cell studies. Cell line culture. For this purpose, we used oral epithelial cell line H400 which was provided by Professor ILC Chapel, University of Birmingham, UK. Cells were maintained in DMEM, supplemented with FBS and L-glutamine. Cells were incubated at 37 degrees centigrade and 5% CO2 in CO2 incubator for 24 hours. After this cells were treated with trypsin and transferred to 12 well cell culture plates. Cell were cultured and 12 well cell culture plates. A scratch was generated using 20 microliter plastic piper tape, which created actually 1 millimeter of wound, and then the extracts each well was treated with plant extracts at different concentration or positive and negative control. For positive control we used platelet derived growth factor, and for negative control we used PBS. So we used three different concentrations: point 5 milligram per of dried powder per ml, zero point 0.5 milligram of dried powder per ml, or extract of dried powder per ml, or 0.05 milligram of dried powdered extracts per ml. For statistical analysis, we used this images software to analyze the image which we captured after different intervals. That is immediate after wound creation, 4 hours after wound creation, 8 hours after wound creation, 18 hour of after wound creation, and 24 hour after wound creation. And then we performed HPLC analysis to identify and quantify the polyphenol A contents. The potential cell proliferation ability of H400 of Sigillum aromaticum extract was evaluated at 0, 4, 8, 18, and 24 hours. Three different concentrations, as I have already discussed: 5.5 and 0.05 milligram of dried plant material per ml were tested. The wounds, how, this is how the wound were at the initial stage, immediately after wound creation. Um, we have treated with three different concentration. Then we have positive control, and then in A, and then we have negative control in E. After 4 hours, same. In A we had positive control. In B we have 5 milligram per ml. In C we have 0.5 milligram per ml of extract, and D we had 0.05 milligram per ml of extract, and in E we had negative control. So the healing process or cell proliferation process has already started. After 8 hours, the contraction was there, but it was difficult, or the differences were not very clear. But after 18 hours, after 8 hours, this is how the wound contraction percentage was. There was differences, but at 18 hours, the differences were more clear. You can see in positive control, there is healing, but still less compared to the different concentrations of plant extracts. In plant extracts, the the highest concentration and the concentration that is zero point 0.5, second low concentration, that is 0.5 milligram per ml, have more healing than compared to 0.05 milligram. And in negative control, the healing process was even slower. So you can see there was significant differences after 18 hours between positive control and highest concentration positive control, and second lower medium concentration. The lowest concentration was not significantly different from negative control, and significantly lower than positive control, whereas the higher concentration, that is 5 milligram per ml or 0.5 milligram per ml, were significantly higher than the positive control, and of course significantly higher than the negative control. And after 24 hours in positive control and all the concentrations of plant extracts, there was complete healing. So under optimized condition, the proliferation We can summarize that under optimized conditions, the proliferation ability was highest at 5 milligram per ml among tested concentration of Cesium aromaticum. Estimated estimation after 18 hours produced more wide-ranging results as compared to 4 hours, 8 hours, and 24 hours. In HPLC analysis, we have identified three major polyphenolic compounds that are gallic acid, ellagic acid, and eugenol. The concentration of gallic acid was 225 microgram per gram of dry weight of Syzygium aromaticum flower bud. Ellagic acid was 107 microgram per gram, and eugenol were 96 microgram per gram of dry weight of plant flower egg, powder. So from this study we can conclude that it was determined for the first time that ethanol extracts of Syzygium aromaticum can stimulate the cell proliferation and migration in an in vitro cell scratch assay using human oral epithelial cell line H400. Further studies are however needed to identify the specific compound responsible for wound healing activities. Thanks. If you have any question, you can ask.